My name is Lukas van Zwieten. I am with New South Wales Department of Primary Industries, but I'm also the leader for Program 4, which is currently entitled Integrated and Precision Soil Management Solutions. Okay, a little bit of background on the Soil CRC. So it commenced in 2017, and the key feature of the Soil CRC is that it's a 10-year funding cycle. So we've got essentially $40 million from the Australian government secured for the Soil CRC's tenure, as well as 127 million in cash and in kind from the Soil CRC partners. There are 40 partners in the Soil CRC, including universities, 8 universities, 4 state agencies, and importantly, 20 grower groups. And as far as we understand, it's the largest collaborative soil research effort in Australia's history. So the purpose of the CRC ultimately is to give farmers the tools and knowledge to better manage their soils, to increase their productivity and profitability. So this morning we heard from Program 1 from Catherine Allen, Hannah Beth Luke and Nick Pawsey around investing in high performance soils. That was a very interesting webinar. We've already had Richard Doyle's webinar on soil performance metrics, and I understand Nancy Boland has got his webinar on this time on Wednesday around new products for soil fertility and function. So Nancy will be presenting there, as well as Ehsan Tavakoli on new products for subsoil constraints, and Professor Richard Bell around new amendment opportunities for sandy soils. Today, I'll be just giving a very brief overview of Program 4, and then we've got 2 speakers from Program 4. The first is Professor Terry Rose, and then Dr. Gwen Grelet. Okay, what does success look like if we're amending a soil? And I think this is a really nice example of using integrated solutions where we actually get a positive outcome. On the left-hand side of your screen, you can see a canola crop grown on a tenor soil that has been both delved, so that is bringing clay up from subsoil, and then spaded, so properly mixed. A couple of soil management options there. On the right-hand side, where the farmer didn't do that, his canola crop basically failed. So I think that is a classic example of what success might look like. And hopefully, we can achieve these types of outcomes through the Soil CRC across multiple soil constraints. Again, very brief introduction on Program 4. So Program 4 is essentially split into 3 key outputs. The first output is around novel plant and systems-based soil re-engineering methods. Both of our speakers today are essentially within that output area. And it's around using inter and cover cropping, legumes, biofumigants, and potentially also now regenerative agriculture systems for developing soil biology, soil carbon, and soil health. The second output is novel physicochemical based soil re-engineering methods. And I guess the key driver of this particular output was the fact that most soils have got more than one constraint. And I think we've got some nice projects within that particular output that are starting to understand and develop mechanisms for ameliorating these multiple soil constraints. And I'll give some examples later on of the projects in that particular area. And the third output is around the universal soil re-engineering decision support tools. And I think there'll be some overlap with some of the opportunities with Program 1 later in the life of the CRC for this particular output. But again, we've got one project currently in this output, and I'll give a brief explanation of that soon. Apologies to bore you here, but I'm just going to go through some of these milestones within the Soil CRC. I think it's important for us to set the scene. And essentially what we have on the left-hand side are the Commonwealth milestones, and then across there we have then what those milestones are. And these are the milestones that we need to report to the Commonwealth Government on around the performance of our particular program. And the first key milestone is a test methods for indicators of high performance soils. And I think Catherine's program this morning was talking around some opportunities for the finance industry for understanding what a high performance soil is or what a more resilient soil is. And we've got some projects under there, for example, Moran Rushdie's project at Griffith University for understanding and evaluating soil functional resilience. So for example there, if we're going into a dry period or if heavy machinery is starting to be used, are soils where we've implemented re-engineering, where we've improved soil health and soil function, are they now more resilient to these stress factors? Another key milestone is around rhizosphere re-engineering, and Terry Rose will speak on that in a little bit more detail later on. And what we're also very keen to do now is to ensure that the data that we're collecting, so the data from these particular projects actually gets delivered into output 3, where a lot of our information will be collected and used in machine learning processes or in better decision support tools. So again, it's very much around delivering data into output 3. So the second milestone, which is around the physicochemical amelioration options, we've got Jason Condon leading a project around the mechanistic understanding of soil re-engineering methods. I'll speak a little bit more on that soon. And then Mick Rose has got a project on better understanding herbicide residues and what these herbicide residues might mean as a soil chemical constraint. And there are certainly some very interesting observations that Mick's come up with in the project already. And then we've got a newly funded project, which was a collaboration between GRDC and the Soil CRC, led by Nigel Wilhelm from PIRSA. And output 3, the only project we have in this particular output at the moment, is led by Keith Pendleton. And that's essentially improving the representation of soil productivity and constraints. And he's using the AppSim platform mainly to deliver that project. So I'm not going to take too much of Terry's glory here for his project, but that's a great photograph from Herbert Kane Productivity Services, one of Terry's projects up in the far north Queensland, looking at cover crop options in the sugarcane industry. Again, Terry will probably describe some of this in a little bit more detail. But essentially, so far, there are 5 long-term field trials established across several regions, and he's utilising a soil functional suite of assays. So I think Terry's project has got a good understanding now of what it's trying to measure as far as soil health, soil resilience goes. And there's also a mechanistic summer crop cover trial established at Wagga Wagga. Terry's promised me he's going to give a very nice presentation soon. The project that I mentioned with Moran Rushdie is around evaluating soil functional resilience. I don't know if you guys can see that black box. I'll move that down there out of the way. Evaluating soil functional resilience to stress for developing high performing soils. And essentially here, the general concept is if you've got a more resilient soil, it's probably better aggregated. Due to that better aggregation, you've got more protection of organic carbon. You've probably got less leaching of dissolved organic carbon. You've probably got lower losses of the greenhouse gas methane or less respiration. Whereas if your soil is more vulnerable, it's probably less well aggregated. It probably mineralizes the carbon more quickly, less opportunities to stabilize the carbon. And yeah, certainly CO2 evolution or mineralization of carbon from those soils can be higher. And I guess that's one of the principles that that Moran and Professor Chen's project is working on. And they've got some great data already where they're having a look at the role of compaction as a stress factor for soils. And they're having a look at a range of soils and also amelioration options for those soils. They've completed a 72-day incubation study. They've had a look at a couple of moisture contents with that, and they've had a series of different soil bulk densities, basically different levels of compaction. And I think they're starting to come up with some good indicators of where respiration or carbon mineralization and also enzyme activities spike where soils are more vulnerable. Again, there'll be a lot more data coming out next year for that particular project. Gwen, Grelet is going to be presenting a bit later on in today's webinar. So again, I'm not going to take too much of the wind out of her sails here, but really this project is very much around co-design to understand the role of soil carbon in regenerative farming systems. There's been a lot of talk on carbon in regenerative farming systems, but I think this project here really is around speaking with the growers, speaking with the agronomists, speaking with the scientists, bringing everyone together and designing a project now where we've got a shared vision. And again, I think Gwen will give a very nice presentation there. So Jason's project sits under, this is Jason Condon from Charles Sturt University. His project will sit underneath output 2, which is around the physicochemical amelioration of constrained soils. And they've collected a whole range of constrained soils from the grower group. So it's quite a large project with quite a few grower groups. And we can see the constraints here. They range from nutrient deficiency, boron toxicity, sodic saline and sodic soils tend to feature quite heavily, as well as acidic soils. So again, none of these particular constraints happen in isolation. They're often happening together. And it's quite important for us to understand mechanisms for addressing these multiple soil constraints. What are we best off trying to address? Is it the soil acidity that's causing the key problems? Is it the subsoil sedicity that we need to focus on? So each of those particular items will be the focus of some fairly detailed pot incubation and potentially also some small plot field studies later on in our project. Mick Rose is working on a project called Developing Knowledge and Tools to Better Manage Herbicide Residues in Soil. Now, that top right-hand corner is a fairly typical tweet that Mick has been receiving, and there are lots and lots and lots of growers and agronomists, contacting Mick, asking what's going on. Their crop, in particular, where you might have overspray, or you might be coming out of a particular rotation with IMI herbicides, for example. Some of those chemicals are lasting a lot longer in the soil than they should be. A lot of those issues are driven by weather, so in particular temperature, rainfall in particular, which drives microbial activity, which drives how quickly these herbicides break down. So what essentially we're trying to what Mick's trying to achieve with that particular project is to develop phytotoxicity thresholds for multiple crop and herbicide residues. So while herbicides might be present in soil, in this case there's a picture of clopyralid at Manaaki, and you can see it breaking down in soil over 100 days. It's also moving into the subsoil, but what we don't know is what does that mean for crop toxicity? And the total concentration of a particular herbicide doesn't necessarily relate to how toxic it is in soil. It's all around the bioavailability. And there's some great opportunities in that project to better understand the bioavailability and the risk of these herbicides. And there'll be some models developed out of this project that will assist growers and agronomists. This is a new project. It was by direct negotiation between GRDC, the Soil CRC, and the various partners. That's Nigel Wilhelm in the top right-hand corner. He's from Persa. So the key issues on these extreme carousel soils. So these soils are pH 8.5 and above. They tend to have a very low phosphorus status, low water holding capacity. They also tend to have a high burden of rhizoctonia, poor nitrogen cycling, and severe fertilizer toxicity during germination. And this project will be delivering 4 field sites within the Soil CRC, mainly on the Eyre Peninsula, Upper Eyre Peninsula, as well as in a higher rainfall location in southeast of South Australia. Along with the field trials, there'll be some mechanism studies around trying to modulate the very high pH and also the redox potential of that soil. And because these soils tend to suffer from quite low organic carbon content and low mineralizable nitrogen, we'll be looking at some techniques such as the use of iron hydroxides for stabilizing rhizo deposits. There'll be some unique research within that project. And that project will also have a complementary project sitting to the side of it, which was funded between GRDC and CSIRO to add value to the to this Soil CRC project. And certainly we look forward to Nigel presenting on this project in the future. And this is Keith Pendleton's project. It's around improving the representation of soil productivity and constraints in existing decision support systems. So I think Keith's project, they've had a lot of meetings, and they keep on coming back to AppSim as a platform for delivering these decision support tools. And essentially what they've completed so far is they've prioritized soil constraints within the grower groups that they're working with. They've modeled the fallow water use by weeds to assist in spraying decisions, and they've improved the initialization of soil phosphorus within the AppSim platform. And essentially what this project is doing is creating a foundational model framework to represent physical, chemical, and biological constraints. And there'll be sort of follow-up projects from this one later within the Soil CRC. It's also important to note too that that many of the projects within Program 4 and other projects as well will be feeding data into this particular output, where basically the computing power, a lot of the computing power lies. And success for that particular output might be an app, but more likely it'll end up being modules sitting within AppSim, or maybe How Leaky, for example, that will help agronomists and farmers and decision makers to better prioritize what they're actually going to be delivering within the Soil CRC and beyond. That's my email there, lucas.van.sweeten@dpi.newsouthwales.gov.au. Please contact me afterwards if there are any questions outside this webinar series. But now I'm going to stop sharing my screen, and I'm going to hand over to Professor Terry Rose, who's a Professor of Sustainable Cropping Systems and also Director of the Centre for Organics Research. So Terry, I'm going to hand across to you, and I'm going to mute my screen.
Thank you, Lucas, and hopefully everyone can see and hear me. My name's Terry Rose from Southern Cross University, and I am hopefully about to share with you my slides. Here we go. Thanks again. So I'm Terry Rose from Southern Cross University, and I'm running a project with many on-the-ground partners and numerous research institutions, and it's called Plant-Based Solutions to Improve Rhizosphere Performance, essentially. So what are we doing? We have already established we've already established 5 long-term field trials in different states of Australia. We have one in Western Australia at Facey Group. We have one in North Queensland at Ingham with Herbert Kane Productivity Services. We have 2 in New South Wales, one with Central West Farming Systems and one on the border with Riverine Plains, so border of New South Wales and Victoria, and one in South Australia with Hart Group. So in addition to setting up these 5 long-term field trials, we also have set up some mechanistic small plot trials to try and understand what's going on, and some glasshouse trials, and subsequently some lab studies as well. We've so far recruited one PhD student, Cameron Copeland, and we have a postdoc on the project, Shahnaj Parvin. And Shahnaj is based down at Wagga Wagga, which is roughly in between 2 of our New South Wales farmer groups. And Shahnaj is also running some small plot trials at Wagga. So what are we trying to achieve? We're basically trying to achieve increased plant diversity in these farming systems. And we've got to find, 1, ways to do it, and 2, whether it makes a difference. So in short, what are our management options to increase diversity in these systems? Well, the obvious one, and the one that's used pretty successfully in Australian cropping systems already is rotation. So compared to some other countries, we already have in most of our farming systems what we would consider, or what at a global scale is considered diverse rotation. In other words, we're not growing the same crop over and over again. You could say, basically, if it was profitable to expand those, expand diversity within the rotation, so grow different crops, one could argue that farmers would be doing it already if it was profitable. And to some extent, that may be true. Basically, we can encourage growers to, let's say, grow more legumes in rotations. Unless that's profitable, it's not going to happen. However, there may also be opportunities for us to demonstrate that it may be possible. So possibly there are some barriers as to why farmers aren't adopting a wider range of crops. And there may be some reasons in the soil that this is the case. And for example, quite possibly the acid layer at sort of 5 to 10 centimetres in many red-brown earths possibly is limiting our legume growth, and therefore they don't perform, they don't grow well, and farmers aren't going to adopt them. So in some of our trials, we're actually looking at simply liming and a few other tricks to see whether or not actually rotations would be profitable if we could have the soil in a situation where the crops would perform. That's one option. Another option is to use or get your diversity in the typical fallow period. So that's what we've called cover cropping. And so for most of our farmer participants, our farmer groups in Southern Australia, that would be replacing a typical summer fallow, a weed-free fallow, with some form of plants, putting in cover crops, mixed species cover crops for some period of time before going back into your cash crop. And the last option is to increase some diversity within your actual cash crop. So if we're growing wheat as our staple crop, there may be some options to get some plant diversity within that wheat crop, not necessarily taking them both to maturity. Maybe you can grow some for a small period of time and then take them out. Maybe you want to grow them to maturity, which we call companion cropping. So they're the three key themes that we want to look at. Obviously, different trial sites have different priorities. In Western Australia, it's more of a pasture-based trial. In Queensland, it's obviously a cane trial, which is going quite well. It received a couple of 1,000 millimetres of water on our cover crop trial the year before last, or last summer. But nonetheless, most species survived and performed well. Today, though, I'm going to focus on our cropping systems in Southern Australia as our main focus, which is Central West Farming System sites, Riverine Plain sites, and Hart Group. Central West Farming System site last year was sown, but once the crops were about 5 centimetres high, they didn't receive any rainfall. So that site effectively is going to be resown this year. However, we did get results from Hart Group and from Riverine Plains, and I'll tell you about some of those shortly. So summer cover cropping, what are we actually trying to achieve here? And I've got some photos down below of the summer cover crop at Riverine Plains this year, near Yarrawonga. And as you can see, they're not massive. And that's probably to be expected in an Australian summer where we don't necessarily get a whole heap of rainfall and it's pretty hot. And so our cover cropping systems are going to be a lot different to most of the world. And most of the data that we get pushed upon us from various speakers, and in science, we get papers thrust upon us, it's all, or nearly all, from Europe and North America. And their systems are quite different. And so it's worth us asking, if we want to grow summer cover crops, what are we actually trying to achieve? So if you read the literature from overseas, or if you listen to some of their speakers or see them on Twitter or whatever, one of their biggest drivers is, for example, increasing soil carbon. Now, that works if you grow your cover crop in the winter in between summer cover crops, say in the Northern Hemisphere, where you have plenty of water and you have some sunlight. In other words, you're just adding more biomass to the whole system. So inevitably, you've got across the year, you've got a lot more biomass going in, you're probably going to increase soil carbon. In our system, as you can see down below, we don't grow big summer cover crops. We don't have a whole lot of rainfall necessarily in summer. There's also the chance that any rainfall we do use in summer is going to reduce the growth and possibly yields of the winter crops. And so the question is if we're not actually putting more biomass in throughout the year, but we're simply transferring some biomass to summer that would have been produced in winter, what are we going to do to soil carbon? Are we going to increase it? And the answer is we don't know. Another driver for the Northern Hemisphere is to reduce nitrate leaching. So their winter cover crops have a huge benefit for a lot of the watersheds over there. Question for us, do we get much nitrate leaching over summer? And from the literature, the answer is no, that's not the time we get much leaching because we have a huge evaporative demand and not much rainfall. Reduced erosion. Are we going to get big improvements in or minimisation of erosion by having summer crops? Again, it's a bit of an unknown, and it probably depends on the season, on how much cover you've got from whatever crop you grew last. Was it a big crop? Was it a small crop? What are you doing with your stubble? And so there's a lot of interactions there. Possibly, are we going to get greater water infiltration? Certainly in the north of Australia on the vertisols, there's some evidence that summer cover crops or cover crops in general can improve infiltration in their storms. Whether that helps down south, we don't know. Increased soil health. Well, I think that is probably one of the key drivers. Certainly, if you keep an eye on Twitter or when you hear farmers talk, that is certainly one thing they do talk about, is improving soil function. And I guess as a Soil CRC, that's certainly something we're interested in. And I guess the culmination of all those things, how do they all interact, is going to affect what happens to the next year's crop yields. So, can we expect soil health improvements? Conveniently, someone just put out a big review paper, or it's called a meta-analysis. And for those of you who don't know what a meta-analysis is, it's where you get all the papers that have presented results on a given topic, you put them all together, and you use maths to draw out the key findings. And these guys did that for cover crops and looked at how do they affect the soil microbiome. Once again, it's worth noting that nearly every paper that went into this meta-analysis or the review would have been from the Northern Hemisphere. So they found overall cover cropping significantly increased parameters of soil microbial abundance and diversity by 27%, 22% and 2.5% respectively. Compared to a bare fallow. Now, there's a couple of things that are worth noting here. Firstly, the microbial abundance and the microbial activity that have increased by over 20%. They are typically simply functions of how much carbon you put into soil. So in other words, if you put on, same as if you went and spread manure or you put on a compost, or if you ploughed in a green manure crop, or if you grew another crop, if you increase carbon going in, those parameters are going to increase. So it's probably not unexpected from Northern Hemisphere data that they increase because they're putting more carbon into the system across the year. What happens in Australia if we put a bit more carbon in summer but we lose some in winter? Again, the answer is we don't know. We may get a slight increase over summer. Do we then get a decrease in our cash crop? We don't know, but we'll try and find out. The other interesting thing is the 2.5% increase in diversity. So that, on the face of it, looks pretty minute. That's over. So from all the studies combined, that was the mean or the average increase. The thing is, it's only 2.5%, but it could be a very important 2.5%. We simply don't know. So Cameron Copeland, who's doing his PhD within our project, is actually trying to look at, delve into this deeper and try and work out what are the microbial communities or particular groups of microbes that might have a large effect. So in other words, you might have only increased one of them by, or one particular group by 1%, but it may have a large effect. We simply don't know. And so don't write that off is what I'm saying, and stay tuned. Hopefully Cameron Copeland will be able to elucidate some of that later on. The other interesting thing for microbial diversity is that the time of sampling has a large effect. And so if you sample while you're growing the summer cover crop and compare it to a bare fallow, you find a pretty big difference. And that isn't that surprising really. If you wait, and in our system, let's say we sample before sowing of your cash crop. So we grow the summer cover crop for a month or two over summer, have a couple of months off, and then we go and sow our wheat in May. Is there still a difference when we sow the wheat? We don't know. If there is, does it make a difference? If there isn't, has anything happened that's going to affect the wheat crop, or is the fact that it's died out, does that mean we haven't really got a benefit after all? And the other question is, how long does that persist? So even if there's a difference when you sow your winter crop, is that going to persist through the rest of the season? And again, we don't know, but we've got these trials in and we're starting to take measurements. We've got the summer crop that you saw on the previous page was terminated last week, and we're starting to take these measurements, and I'm really hopeful that in a year we'll have some data. In a couple of years, we'll have a few seasons of data and can start to form some actual robust conclusions based on multiple trial sites and a couple of seasons. So I'll leave summer cover cropping there because while it's interesting, we simply don't have the data. But again, hopefully in a few years we will. Intercropping and undersowing. So this is where we try and increase diversity in the actual growing phase of your winter cash crop. So at Hart Group, the farmers down there were quite interested in actual companion cropping or intercropping, where you grow two species at the same time. You take them through to maturity and you simply separate the seed after harvest. So Piola is a common one, and there's been a fair bit of research globally on that. And as a general rule, you get a land equivalent ratio, which is basically how much land of the monoculture crop would you need to get the yields that you got from your dual crop. And so usually the numbers when you grow Piola globally are sort of 1.1 to 1.2, which means it's much more efficient to grow them together than if you grew them separately. You'd need more land if you grew them as monocultures than if you grew them together. There's been some work in Australia, even in South Australia actually at Minapa, that had similar results. So they found a land equivalent ratio of about 1.1. So in other words, you get more output from your intercropped crops or your companion plantings. However, if you look at the Piola in the graph, we didn't get that. It was roughly 1, and there was a fair bit of error. So actually, in 2 of the replicates, we got a yield advantage from the intercrop, and it was about 1.1 each. But then in one of the replicates, we actually lost yield by growing them together. Why? I don't know. And given that it's pretty consistent across the globe that you should get advantages, we'll have to look into why that wasn't the case in this year. You can see from the picture to the right and from the yields from the monoculture crops in the table, it was a pretty low yielding year last year down at Hart Group. So canola was about 1/2 ton to the hectare. So possibly that impacted, we just don't know. The chickpea and linseed intercrop had a land equivalent value of 0.8, which means it was much less efficient to grow them together than as a monoculture. Why? We don't know, but it's something that needs to be looked into. Whether it's the ratio of seed we use, whether or not a 50-50 split isn't best, whether you're best to manipulate that, whether it's because we sowed parallel rows of each as opposed to mixing the seed in the same row, we don't know. And I guess that's the issue with starting these trials from scratch without a year to muck around and try various combinations, is that we're sort of going in cold. And so we're going to try and run some parallel studies to try and optimize some ratios and that sort of thing while we run these main trials, to try and make sure that when we do put treatments in the main trial, we give them the best chance of success. So the next question is whether or not these intercrops have any effect on next year's wheat crop. So the whole site next year is going to be sown to wheat, and we'll find out whether or not sowing wheat after, for example, a Piola crop has benefits compared to sowing it after a straight canola or a straight pea crop. We've taken measurements of different soil parameters. I haven't actually got the data with me, or it's not presented here because I haven't analysed it yet. But again, it'll be interesting to see what did we change in the soil, if anything. Does that translate to any impact on wheat yield, wheat growth or yield next year? So intercropping, this is back to Wagga Wagga, where we mucked around with growing different species in with the main crop, in this instance wheat, and we didn't—
Terry, 1 minute please. Sorry mate.
We didn't take these crops to maturity, so we spray them out at different times. So that's a vetch and wheat crop, and basically if we take the vetch out after 8 weeks or before, it doesn't affect wheat yield. On the left is actually not the data from that picture. On the left is the data from Riverine Plains site where you saw the summer cover crops. They're the 2 wheat lines over on the right. So the winter crop was just wheat. However, there's a subclover wheat plot right in the middle where we had subclover growing with the wheat for a period of the growing season, and it was sprayed out. And you can see it didn't cause a yield loss. Again, the question is, does it have any effect on the next crop? So next year we sow that site to canola and see whether having something growing with your wheat made any difference. Finally, we've got a few mechanistic trials at Wagga. We're trying to get a look at these summer cover crops to find out what the differences are in their rooting growth patterns. For example, we probably don't want something that goes too deep and mines too much moisture. What's their growth habit like? And beyond that, we're also going to do some pot and lab studies trying to work out how much carbon are they actually allocating to their root exudates, what are their root exudates, and try and get an understanding of the different, the possible benefits of individual species so we can possibly tailor our mixes later on. That's all I've got to say, and I'd like to thank our project partners. And with that, I will sign out and hand it back to Lukas.
Thank you very much, Terry. And hopefully we'll have a few minutes at the end to take the Q&A. I'm going to hand over now to Dr. Gwen Grelet, who is a plant soil ecologist with the Manaaki Whenua Landcare research team in New Zealand. So thank you, Gwen. I'll hand over to you.
Thank you, Lukas. So it's Gwen Grelet from Manaaki Whenua Landcare Research in New Zealand. So I'll share my screen and hopefully this will work. Can you team confirm it's working?
Yeah, it looks great, Gwen.
Yes.
Thank you.
Looks great. Okay. So I will just give you a rapid overview of the Soil CRC funded project, which aims to be a pilot project studying regenerative farming system and more specifically understanding the role of soil carbon for the good functioning of this system. I'm the project leader, but this is a collaboration. And let me see if I can, yes. So we have multiple partners. It's a collaboration with David Davenport and Claire Donnelly and Amanda Chappell from Percy, with Felicity Gilbert from Whitbell and RM, Kirsty Yates from Soils for Life. And we're also collaborating with Nicole Masters from Integrity Soils and Dr. Will Allen, who is based in New Zealand and is developing various tools and mediation processes and facilitation processes in the theme around sustainability. So very appropriate for our topic. And roughly my talk, what I will do is, our project is 2.5 years and we are just finishing the 7th month. So I will give you a brief overview of background on what is regenerative agriculture, why do we want to research it, and where is the science so far. And then I'll give you an overview of our project and where we've got to at the moment. So what is regenerative agriculture? And that's the $1 million question. A lot of people that are wanting to work in that space and talking about it want to come up with a definition of it. So a definition was proposed by the Carbon Underground and CSU Chico, so from the United States. And talking about regenerative agriculture as a management that, in a type of agriculture that describe farming and grazing practices. Let me see, I can't see which has multiple benefits. Amongst these benefits, reverse climate change by rebuilding soil organic matter and restoring degraded soil biodiversity, which results in both carbon drawdown and improving the water cycle. So that those are terms that are very easily to understand and somewhat also very challenging for the scientific community as well. The other part of the definition is that regenerative agriculture is a holistic land management practice, which means that it looks at it with looking at the farm as a complex whole ecosystem and seeking to leverage the power of photosynthesis in plants to close the carbon cycle, build soil health, crop resilience, and nutrient density. So in other words, regenerative agriculture seeks to minimize the synthetic agricultural input and instead rely on biodiversity-driven processes. So maximize this biodiversity both at field and farm scale and maximize carbon and water capture. There is also, there's multiple definitions that are going on. And as Terry just explained earlier, A lot of these concepts come from the Northern Hemisphere, North America, and for this one also from Africa, South Africa. And the ecosystem are very different. And so there's a need to adapt the definition and what we mean by regenerative agriculture to the particular Australian context. So definition proposed by David Marsh, a leading regenerative practitioner and educator, is that regenerative agriculture is about managing to allow measurable improvements over time in ecosystem function and biological diversity, driven mostly by the self-organizing ecosystem towards a social, ecological, and financial goal. So I would like to attract your attention to the term self-organizing ecosystem, which means that the management seeks to look at the ecosystem as a complex adaptive system, which means that the system will have emerging properties that we cannot predict from just studying all of the parts of the system on their own. And that's a challenge for scientists like myself, for example. The other challenge is that regenerative agriculture is defined on the outcomes that are thought rather than the practices. Unlike, for example, when we talk about organic agriculture, the definition is mostly based on the practices and what we are allowed to do or not allowed to do. So what does it look like? So these are photos from Charles Massey, a prominent Australian regenerative practitioner, and you can see that within a decade, using adaptive grazing management and other soil mineralization processes, Charles has managed to restore the vegetation on his property. And then another example is Stuart McAlpine in Western Australia, who was producing grain, and you can see that under very challenging environmental condition, Stuart is still managing to produce extremely good soil condition, very good rhizosphere with a lot of soil sticking to the roots, indicating quite a lot of life around the roots. And he's a very successful farmer in terms of production. So why is there so much interest in terms of, well, research for me, for example? Regenerative agriculture is starting to be really talked about in the press. So I've pulled up some articles that were released in the press just looking at Australian regenerative agriculture. And what is quite striking is that regenerative agriculture is seen and almost advocated or claimed as having a lot of potential benefit, such as mitigating climate change, reversing the decline in farmers' wealth and well-being, reversing the decline in biodiversity, and potentially being a pathway forward to restore water cycle and even recover from fire, which is very timely. Also, there's a huge interest from a social standpoint because the farmers are organizing themselves. There's a powerful groundswell, which is actually global, not just in Australia. It occurs in New Zealand, the UK, Europe, the Americas, Asia, Africa, pretty much everywhere. It's been discussed at the UN. It was discussed at the last COP25 summit. There's a huge interest by impact investors in investing in regenerative agriculture. This is a new platform launched 3 weeks ago in New Zealand, channeling investment to promote the adoption of regenerative agriculture in New Zealand. And then very recently, amidst the crisis that is affecting all of us here in this world, in the entire planet, COVID-19, there's now opinion papers. Some are anchored in scientific facts, some not, but really discussing is the situation that we're experiencing now a result of us destroying biodiversity, or for example, promoting too much factory farming. And so regenerative agriculture is really moving away from factory farming and is seeking to increase biodiversity on all levels. Right, where is the science of regenerative agriculture? There isn't a lot of papers so far being published. There are papers that are individual studies that are looking at particular practices that are adopted within regenerative farming methodologies, but studies that have looked at the whole system without distinguishing each particular practice and just looking at the outcome at an ecosystem scale are far and few between. So I'll highlight one study that was picked up by the journal Forbes, so it made the front page in just 2 months ago. This was the study of the benefit of regenerative agriculture for producing corn in the coal belt of the US. And the study is led by Jonathan Lundgren. And basically what they've done is compare regenerative farms with their neighbors that were using the best practice of the region. And they defined the regenerative farm as those that had adopted at least 3 out of 4 typical regenerative practices for that particular production system, such as multi-species cover crop, no insecticide, no tillage, and integrating grazing. And what they're showing here, you can see on the chart, the revenue and cost for the conventional farms versus the regenerative farms. And in white are the profits. So you can clearly see that there is more profit in regenerative farming system. And in colored and are all the different costs that are associated with the production of this corn. And you can see that the profit is due to the reduction in cost, but also the increase in profit, which is due to increase or maintain production, or also increase financial benefit per unit biomass produced. So from a financial standpoint, the regenerative farming system makes sense. In terms of coping with pests, whilst the regenerative farmers didn't apply any insecticide, the team showed that there were very much less pests recovered, in fact, by an order of magnitude in regenerative compared to the conventional. And then there was a really good relationship between soil health and the profit. So you had indicators such as bulk density or % organic matter. So the organic matter, was its increase on each of the property, then the profit for that property increased. What are the evidence from Australia? There's a really nice paper. This is a white paper, but it then made it into another bigger paper, peer-reviewed and published in an academic journal, led by the University of Sydney, and is describing and studying the processes at landscape scale on colony-sized property. It was also featured in the film 2040, recently released. Colin has invented a method which is called pasture cropping, where he's basically direct drilling and sowing grains into a pasture of a perennial native grassland. And he's then integrated livestock grazing inside of this. So he's stacking enterprises. And what they showed the team is that calculating using various indicators, the nutrient cycling index on Colin's property, which is the column A on your chart on the top right, scored higher than the comparative neighbor, which still had perennial grass patch, but not as many, and didn't deploy the same type of management practices. So now what are we doing with our project? So I will go over briefly our approach and methodology.
Gwen, sorry, Lukas here. Could you wrap up in about 3 minutes? Sorry about that.
Absolutely, yes, we're on track. So our project is organized in 3 parts. The first part is where we are just about to finish, is our part 1, which is set up our co-innovation platform. And what it means is that we are basically defining the research question and that we are going to ask, and the hypothesis that we would test together with farmers, agencies, scientists. So that is actually quite an involved process, because everybody comes from a different knowledge system, different culture, different way of working. After that, we will be taking the research question, hypothesis that we want to test, to draw a research plan and then implement it, which will be the part 2. And then in part 3, we are evaluating our whole process. So I will briefly describe the activities that we've been undertaking since the start. So roughly, basically, we are only finishing the part 1. So our activities started from looking at what is the space we are functioning from, and what is our culture, and what do we want to achieve with this project within the core project team and our contractors, starting with a value and needs analysis, all the way to how do we get a set of indicators and an experimental design. and I will just unpack this. So we first started with a meeting in August. Because our project attracted quite a lot of attention, we were even gifted a meeting room for free. We did some value and needs analysis so we could develop a shared vision. And from that, we started to build a strategy for our co-innovation. The first thing we did was to appoint a research planning group, which is composed of 10 prominent regenerative farmers. Some of which you know, for example, Cole Zeiss and Stuart McAlpin and Nicholas Kelly and so a few others. And then what we did is we guided everybody, the group, to come up with questions that could be asked through a research project, organized in 3 themes. What question can we ask to better understand how regenerative farming systems work? Second theme was, what question do we need to ask to provide the information that will help farmers decide whether they want to adopt regenerative agriculture or not? And then the third question was, the third set of questions we wanted to capture were, how do we study the role of soil carbon in regenerative farming system? And at the bottom of the slide, you can see the word cloud from all the questions that were submitted by the group. We then deployed a theme analysis, which is a classic social methodology. So this is the example of what we did with the questions submitted for soil carbon, how to study the role of soil carbon. So on the left, you have all the questions submitted that we grouped by theme using a coding and then organizing all of the questions based on that coding. We then came up with a major theme, and then on the top right, you have all the themes, the main themes that are associated with what do we need, what kind of question can we ask to understand the role of soil carbon in regenerative farming system. And that then led us to a set of indicators and experimental design, which is in the next slide. So we now have come up with an experimental design where we're going to compare regenerative farms with their neighbors that implement the best practice recommended at this point in time. But we're also designing our experiment in a way that we are increasing the baseline knowledge on regenerative farming system. We've also come up with a design to how we're going to sample at the farm level to capture the spatial variability and the temporal variability. We've come up with criteria for which case study are we going to include in our study, and how we're going to select the sites that we actually sample for the environmental parameters. And then here is a whole bunch of indicators that we are considering and that we'll be choosing based on budget and feasibility and when we will be allowed to go sampling on site due to what's going on with the epidemic. And that's it for now. This was already quite a lot of work. So we have engaged with quite a few people. We have 6 organizations in the project team, and farmers, and all their network, and the guys that we are working with are highly connected. They themselves have companies, are educators, so there's a huge reach out. We're collaborating closely with all the people based in New Zealand through the collaboration with Manaaki, Whenua, Landcare research. We're collaborating with Dr. Annabeth Luke from Southern Crown University and Lukas van der Zwieten from New South Wales DPI for some of the carbon analysis. And we are starting to build some collaboration with the Ministry of Primary Industry and the National Science Challenge Our Land and Water in New Zealand, who is also developing some work on deriving a suite of indicators for studying regenerative agricultural farming system. And that's me now. And if you want to contact me after the question, I've put my email address at the bottom of this slide. And thank you very much.
Thank you very much, Gwen. Great presentation. We have got a stack of questions here and only about 4 minutes left to get through them. So I'm not going to do them in order. I'm going to do some questions that might answer a few in the one go. So this is probably a question both to Terry and possibly to Gwen as well. Is there any evidence that maximising plant biodiversity will maximise soil carbon and water capture? So I don't know, Terry or Gwen, which one of you would like to take over on that one?
I'm happy to take that one.
Thanks, Gwen.
Yeah, so I think, Terry, you can answer that for a specific Australian system, but throughout the world, there's been many, many experiments. The most famous experiment is the Jena experiment in Germany. It's about 20 years of work, fully controlled by scientists. And as you increase the number of species from 1 to 60, you almost have a direct linear relationship between the number of species and the amount of carbon that is stored in the system below ground. And what do you say for Australian system, Terry?
For Australian systems, we have zero data, but we have some experiments in place to have a look at it. But there's also, in addition to that European study, there's also other studies that aren't so conclusive from around the world. So yeah, there's certainly some papers that do suggest that, but there's also plenty that suggest that it's not quite so simple as maximising species. If you pick 3 particular species, you might get there. And there's also the question as to whether maximising carbon below ground is simply a function of maximising your total carbon of the crop. And quite often, if you plant a mixed species, you do end up with more biomass than individual species. And often the reason is, well, in some years you'll get an individual species will yield higher than a mixed species, but over multiple years, the mixed species tends to be up there all the time. And the reason is because in different seasons, different plant species do well. So if species A does well in a wet year and doesn't do well in a dry year, well, you're in trouble one year. By having a mixed species, you tend to have everything covered, regardless of the season. So on average, over a 10-year period, you'll tend to have got more biomass above ground and therefore below ground from a mixed species cover crop. But again, there's always exceptions. And again, from Australia, we have no data. So we're going to try it in cane systems this year, and at Wagga, we'll set it up this year as well. We can't do 16 mixes in every permutation of that, but we might have some monocultures, some bicultures, a couple with 3 or 4, and then one with 10. And we'll just see, we'll track it over the long term and we'll see what happens to soil carbon. So I know that doesn't answer questions for Australia, but we'll try and find out.
Thanks, Terry. And look, there's certainly been quite a few questions based around the influence of soil moisture availability and carbon capture. So I guess that's a part of your project and potentially Gwen's project as well. How does drought influence carbon capture and the actual work that a farmer might do around cover cropping, for example? So any more comments on water or drought, Terry?
I saw there was one question about how does it affect temperatures versus standing stubble, which is that also links into water because presumably there's going to be an interaction with stubble. So let's say if you've got a really dense stubble cover, you've come off a big year, you've retained all your stubble, then particularly towards the latter end of summer, any water that falls, you've probably got protected from evaporation. Whereas if you've got minimal stubble cover, probably the top 10 centimetres at least of your soil profile is going to evaporate. And so in that instance, you can presume if your cover crop only mines that top 10 centimetres and doesn't go too far, yep, you're gonna have saved water and you're probably going to minimise temperatures. But if you've got dense stubble cover, probably doing the same thing, maybe there's no point. And the only one thing we have noticed of when we've sowed our summer cover crops, and I've spoken to a few people who've haven't published anything, but they've trialled them. You do tend to notice some of your stubble, so you lose a bit of stubble cover in the actual sowing process of sowing the summer cover crop. You sort of disrupt that stubble cover. And so there's all sorts of interactions. And again, hopefully in a couple of years, we'll have some answers.
Thanks, Terry. Look, conscious of time, it's 3 o'clock now, so we need to wrap up. I understand that we will be able to respond to a few of these other questions offline, and I hope that we can all help out with answering those questions as well. And just a reminder, this is for the Soil CRC participants, that same time tomorrow, 2 p.m. Sydney time, we'll be doing a milestone review for Program 4. So I hope as many of the Soil CRC participants can make it along to that. So I'd just like to say thank you again very much to all of the attendees today. I hope you found the presentations useful, and yeah, thanks again.